The Covid-19 pandemic is changing the whole world. Lockdowns have occurred in many countries and over 1 million people have died from the infection. Infectiologist Professor Lübbert knows the crucial points of the infection.
In the beginning of the disease, we can't predict whether it goes into light, moderate or severe disease. So after day 6, 7, 8, there is a switch from light or moderate to severe disease. We can't predict so far. So it would be very nice to have a diagnostic tool for prediction before this stage of disease.
A new test could be the turning point. Professor Mischak from Mosaiques Diagnostics in Germany is the developer of the proteome analysis method.
This urine-based proteome analysis enables an early detection of severe cases, and this, of course, at the same time also enables an early intervention.
In the proteome analysis, up to 1,500 different proteins and peptides are measured from a patient's urine sample in order to be able to identify diseases at a very early stage. The method is already being used successfully for some tumors, cardiovascular and kidney diseases.
In principle, chronic diseases, or almost any disease, develops on a molecular level. First, there are molecular changes which, if they are not affected, will ultimately result in damage to the organ and then ultimately result in clinically relevant disease. A detection at an early point in time at the molecular level enables an early intervention.
The patients of the kidney specialist, Professor Beige, already benefit from the early detection.
Using the urinary proteome analysis, we could be able to differentiate and diagnose certain kidney diseases. And because of the relationship between the endothelial aspects of kidney diseases as well and COVID-19 on the other side, we got the hypothesis to test that value of proteome analysis also in COVID-19.
The proteome analysis has already been used successfully in more than 100 COVID-19 patients worldwide to predict which patient needs intensive medical care and which does not.
So far we know that the ACE2 receptor is responsible for entry of the virus into the body and into the cell, and the replication in the cell starts, and cell destruction is a sequelae on the local level. And also, as soon as the virus enters the human body, an inflammation is triggered and immune reaction is triggered. And if this kind of immune response goes out of the normal regulatory cycle, you get hyperinflammation, a cytokine storm, the worst case, and clinical problems, severe disease or even death of a patient.
The proteome analysis, therefore, pays close attention to changes in the endothelial cells, the cells that are first attacked by the virus.
By now it is very well established that COVID-19 results in endothelial damage. The endothelium is basically the inner side of the vessel. These massive damages to the endothelium, of course, also result in changes in the protein in the blood and ultimately in urine.
We were able to associate the severity of the disease with nearly 100% prediction accuracy along with the urinary proteome.
The early detection of the course of the disease in COVID-19 patients can make it easier to deal with the pandemic worldwide.
I mean, it's a potentially simple diagnostic tool. Just collect urine, give it in a standardized procedure in a special lab, and wait for the results. Quite easy. So I think it would definitely help to organize clinical care.
By performing this type of diagnosis, we can predict which patients will have a severe or critical course of disease, which patients will undergo a moderate course of disease. We can initiate personalized treatment, and we can prevent, at least in many cases, severe disease or even death.